Thank you very much for the kind introduction. Do you hear me fine?
Yes.
Okay.
Very well.
Yeah, it's great to see so many of you online. It's amazing. It's almost 500 participants. That's great. Yeah, we'll be talking mostly about single cell spatial genome organization with some epigenetics in there. So our lab is interested to understand how 2 meters of DNA is organized within the confines of a small nucleus, and how this organization contributes to the regulation of nuclear processes with an emphasis on the regulation of gene expression. We're interested to understand specifically how spatial genome organization contributes to embryonic development. We do that in mouse fish systems. Okay, so these are the systems that we developed over the years. And if you're interested in hear more about any of those, you can contact me. But today I will, at the end of the talk, I will only be talking about this system where we developed a conditional mouse in which we can map spatial genome positioning in any cell of the developmental mouse. And this is a nice collaboration with the Mootloos lab. So just by looking at the nucleus, and this is an electron micrograph of the nucleus, you can already learn a lot about how the genome is organized with this densely packed chromatin over here and the more loose type of chromatin, which is more localized towards the nuclear interior. From this, you can already tell that the regions at the periphery, which are over here, these genes are likely to be in an inactive state, whereas the regions here in the more open type of chromatin are more likely in an open state. Yet what you don't know is what the identity of those regions is that are positioned here, as opposed to the regions that are positioned here. And you also don't know how this organization changes in different cell types, how it's different in different cell types, and how it changes in the course, for example, of development. So how do you identify the spatial genome positioning? And especially in such a way that you obtain single cell resolution, hence the reason why I'm in this section. So just an analogy to a method that everybody knows, chromatin immunoprecipitation, we use a technique that's called DamID, and I'll just explain, I'll just take a few slides to explain it because not everybody knows this technique generally. So in analogy to CHIP, with DamID also, when you're interested to know where your protein of interest sits on the DNA, with CHIP, you fixate your cells, and then with a specific antibody, you pull down your protein with the associated piece of DNA where your protein sits, you sequence that, and therefore you identify the regions of the genome where your protein is located. With DamID, basically, you have the same objective, instead that now you express a fusion protein of your protein of interest, again here in green, fused to a molecular stamping mechanism, which is this enzyme over here. So it's an in vivo method and it doesn't involve any fixation. And to go into that in more detail, the stamping mechanism is this protein, which is called DAM, and DAM stands for DNA adenine methyltransferase. So as the name implies, it methylates adenines in the DNA within a GATC context, and this is unique to higher eukaryotes. So in order then to understand where your protein of interest localizes to the genome, you fuse them to your protein of interest. Your protein of interest then localizes, you express this in vivo, your protein of interest then localizes to the genome, and any DNA in vicinity of the landing location of your protein will receive this methyl print in the form of this methylation on the GATC. So effectively, you create a blueprint on the DNA of the location where your protein sits. So I told you we're interested in understanding spatial genome organization. So how do we measure this? And this we do by fusing them to a protein which is called lamina-B1. And we chose this protein because it exclusively lines the inner nuclear membrane here in this thin layer. This is intermediate filament meshwork that is only about 20 nanometers thick. You find it exclusively here. So this is lamina-B1, and any DNA that would then contact the nuclear lamina will receive this methyl print in the form by DAM in the form as methyl groups. So any DNA that was here that I showed you previously in the electron micrograph will be captured with this technique. So to illustrate that further, you would have to introduce a fusion protein of DAM fused to lamina B1, introduce that in the cell because it's an in vivo method. Lamina will direct the DAM protein towards the nuclear periphery, and any DNA that will touches the nuclear periphery will receive this methyl print. Then the DamID pipeline, genomics pipeline, is extremely simple. Everybody can do it with some molecular biology background. You simply isolate genomic DNA, which then of course has the methyl groups at the positions where your protein was located in your cells. You digest with DPN1, which exclusively cuts the methylated, the regions of the genome that are methylated by the interactions with your fusion protein. And then you ligate double-stranded adapters to these fragmented parts of the genome, and you can selectively amplify them and sequence, and therefore identify the regions where your protein was located. So for a fusion to lamina B1, this is what it typically looks like. What you're looking at is an interaction profile of a human chromosome 17. And typically what we observe in this case is that there are these big regions, the big domains, which we call lamina-associated domains for obvious reason, that we find associated with the nuclear periphery. So for this specific chromosome, we see that this chromosome is associated with the periphery with these two big domains on the right end of the chromosome, and this interaction here on the left end of the chromosome. Now, this has been done for many, many different cell types and many different species. And typically for mammalian species, you find about 1,000 of these lamina-associated domains. They're really big, so they range from 100 kb to 10 MB in size. They contain thousands of genes. And as you could already guess from the electron micrograph, they are generally lowly transcribed. So this we could, of course, confirm now also by genomics. And this is important to remember, is that these LADs cover about 35 to 40% of the entire genome. And that is true across species and across cell types. Okay, but why does single cell DamID now work so well? Why does DamID work so well for single cells? And this is because it's an in vivo method. So it doesn't require any tissue handling, because we do the molecular mechanism is the stamping already in vivo. So the blueprint is laid down in vivo. And then you simply just have to capture your cells and deposit that into a container. The container typically in our lab is a 384 well plate. And then what you only need to do is these enzymatic steps that I just explained to you. So you deposit your cell into the container, you lyse and you cut with DPN1, then you ligate your adapters and you amplify in a certain way, which can be PCR or linear amplification. So there's very little tissue handling involved. And what I just want to emphasize in this case is that what is different from DamID from other methods that you probably know, like cut and tag, is that it doesn't require any input material. So with cut and tag, you require thousands of cells to do your tissue preparations on in order to obtain a few hundred single cell profiles. With DamID, you just need one cell to obtain one profile, which of course has some advantages if you work with tissues that have very low input starting material. So this is kind of what we developed over the years. We developed single cell DamID already in 2015, which was the first method to map protein DNA interactions in single cells. Then we extended this method to also include transcriptomic readouts, so we can capture these two modalities from the same cell. Very recently, when we just submitted, and you can probably find it on bioRxiv today or tomorrow. I just submitted it this morning. EpiID, and this is not correct. EpiDamID and T, where we can capture histone modification profiles of many different chromatin states together with transcriptomics, which obviously is not possible with DamID. And we developed a method where we can capture two protein binding events in the same cell, or histone modification and binding events. So I will go through almost all of them sequentially, starting chronologically with single cell DamID. So this is just an example of what you obtain with DamID. What we see is, I showed you already this interaction profile of the average, where we have these three prominent domains. If you look across single cells, and I just showed you, I'm showing you three single cells, you see basically this where in general, the domains are maintained, but we see large variation between individual cells. So this particular cell is associated with the nuclear periphery, again, with these 3 interaction domains, whereas this cell only has these 2 domains on the right arm of the chromosomes that are associated, not the left arm. And then this 3rd cell here has its chromosome only attached with a small interaction domain here on the right end. The rest of the chromosome is somewhere located towards the nuclear interior. So from these type of profiles, what we observed is that we see a lot of cell-to-cell variability. For example, here we stack 91 cells on top of each other, which you're looking at here as a heat map. So the dark shading here indicates interactions with the nuclear periphery in this particular cell, and every horizontal line is an individual profile. So what you can observe here is that these 2 prominent domains, you find them associated with the nuclear periphery in most individual cells, not in all individual cells. And also when it's not there, you generally also find the domain next door and not to be there. So there is some coordination also in the associations. Whereas here, for example, this chromosome associates with the nuclear periphery much less frequently. And also the domain structure is a bit interrupted over here. So you can imagine you can do a lot of fun things with these types of data sets. But this data has been published already a long time ago. So if you're interested in looking into that, please have a read on the paper. But I will focus during this talk more on what we think may be the function of LADs. So we think it has a structural function, and that was work that we've done previously. But what's the function of LADs in gene regulation? I showed you that these regions at the periphery are in an active state, but what is the contribution of LADs to keeping these LADs or making sure these genes on the periphery are in an active state? So for this, we developed single cell DamID, and the T, of course, stands for transcriptomics, to capture both the spatial genome positioning together with transcriptomics. So we can get these 2 modalities from the same cell, and then we can start to see what the implications are of this single cell heterogeneity for expression output. So I'll show you some new data. This is in ES cells. Also to illustrate that actually one very nice side effect of this single cell DamID method is that also the quality of DamID profiles improve significantly. So not only are we able to capture 2 modalities from the same cell, but also only the one modality, DamID, improved a lot. As you can probably appreciate here from these 5 randomly chosen interaction profiles on chromosome 1 in ES cells. And to further illustrate that, I'm just showing you here a single 384 well plate. And I told you we do these as 384 well plates with robotics. And this is just all the cells without filtering. So as you can see that all the cells that we returned after sequencing, they gave actually very high quality single cell DamID profiles, except for these cells over here, which don't give a profile. And also in ES cells, as compared to what I showed you before, which was in human cancer cell lines, is we see the same type of domain organization in single cells, and that we have small domains that have a lower contact frequency, so they associate with the lamina less frequently. We have these very big, large domains that associate with the periphery in almost every individual cell that we analyze. And then we have these interesting domains here, which are asymmetric in that we call them asymmetric in that they have very sharply defined borders here on the left end. And then there is this less defined border on the right end, which you can call furry or fuzzy borders. And we're very interested in looking into this in more detail. And we hope to understand by looking at this systematically, what defines a border of a LAD. Then, of course, what we can do, because we have the 2 modalities, is a very simple analysis where you look whether the heterogeneity we find in the spatial positioning of the genome in relation to the nuclear periphery, what the impact of that is for gene expression. So what we've done here, we asked the question whether the genes that are located within this LAD, for example, whether when it's associated with the lamina in one cell, whether the repression of those genes is increased over the same genes in another cell when this region is dislodged from the nuclear lamina. So we simply calculate a log-fold change in expression between the regions that are associated with the periphery in cells, as opposed to the same regions in other cells that are dislodged from the lamina. And of course, the same over here. And we do that in genome-wide. And indeed, what we find is that in general, the expression of genes is lower for genes when they are positioned at the nuclear periphery, as compared to the same genes in other cells, individual cells, when they are dislodged from the nuclear lamina. So we think this is a very important point, in that the differential positioning of LATs that we find across many cell types has actually, or is at least associated with changes in gene expression. So we think this might actually provide a stochastic framework for cellular state transition, because you can imagine that every individual cell has a different set of LATs that are dislodged from the nuclear lamina. And now we find that that is actually associated with differences in transcriptional outputs as well. So we think this may provide cells with an opportunity to change their transcriptional identities, for example, in developmental trajectories. So this is something we're actively investigating currently. But to illustrate this point a little bit further is that we do not only find this heterogeneity in positioning in tissue culture cells, or ES cells is what I showed you, and these cancer cell lines, but also in all the cell types that we looked at so far, also in vivo. So here's just an example of the 2-cell stage where we developed the protocol previously with the Torres Padilla lab to obtain single cell lab profiles in pre-implantation development. And again, I hope you can appreciate the large variability that we observed in lamina association, even between the 2 cells of the 2 blastomeres of the same embryo. So again, this is the big advantage of DamID, is that we can simply capture by mouth pipetting the cells that we're interested in. And here we specifically captured 2 cells that belong to the same embryo. And we see extensive heterogeneity, similar to what we've observed in ES cells. Interestingly, when you actually look at other important features of gene regulation, we looked here at K27 trimethylation and chromatin accessibility, which we do with an untethered DAM enzyme. We do not find this variability. So at least at the 2-cell stage, yes, we see a lot of changes in chromatin in spatial genome positioning, but that's not associated with heterogeneity in other chromatin features, at least not the ones we looked at. But of course, we're interested, this is a 2-cell stage, and 2-cell stage the transcription is just turned on in these cells. So it will be more interesting to look at 8 to 32 cell states, where the cells are start to take their first decision to become either part of the embryo proper or be part of the extra embryonic material, the supporting material of the trovectoderm. So we're actively looking into that. I just want to say, if you're interested to know how we created those profiles, although they're not in the paper, but the strategy behind it, please have a look at bioRxiv tomorrow, probably, where we have deposited this paper. So take-home message is that heterogeneity in spatial positioning is associated with differences in gene activities. And we think this may be important, especially in developmental processes. Okay, so what is the function then of LATs? We've seen this heterogeneity. And we've seen that the genes in LATs are generally inactive, but is this cause or consequence? And by no means I'm going to give you the answer, but this is actively investigated in our group. But I think we're starting to develop the tools and the model systems to really start addressing this. So I will show you 2 examples for the final part of my presentation. So here we actually took advantage of a very nice, quite well-defined process of cortical development. And here we teamed up with Onur of the hospital next door, who's an expert on this. And we wanted to take advantage here of cortical development around these embryonic stages. And what's happening here is that the apical progenitors will gradually differentiate into all the different cell types that host the mature cortex. You have this very nice, interesting differentiation trajectory with known transcription factors involved in this process. So we wanted to take advantage of this well-defined process to understand how spatial genome positioning ties in with the regulation or gene regulation during this differentiation trajectory. Of course, in order to do so, we had to introduce our DamID system. As I told you, DamID is an in vivo method, so we had to introduce this into the living mouse brain. And of course, this was an expertise that Onur brought in. So what we did was actually electroporate the pups. So you take the pups from them out of the mother, you electroporate the brain, so especially the cortex, to introduce the DamID markers in there. Then you place the pup back into the mother and allow the pup to develop further for 2 days. And this was done at E14. And then we capture the cells when they've just started differentiating into neurons and glia for single cell DamID. We can capture the cells based on the expression of fluorescent markers. We sort that into 384 well plates, as I told you before, and we can perform DamID and T. Then because we have these two modalities, the mRNA and the DamID, we can, based on the mRNA, determine the cell types. This is probably very well familiar. This UMAP to you. This is this two-dimensional representation of multidimensional data, and the dimensions are here, all the transcripts that the cells produced. And here you have all the different cell types that are annotated. We have 7 over here. And then based on the well-known markers, as I said, this is a very well-known system. We can determine what these cells are. So the apical progenitors are known to express these markers. They're expressed over here in this TIL. So we know this TIL represents apical stem cells. Same here for the basal progenitors. They express these markers. So we know these are progenitors and these are the neurons. Now, we of course do not only have the mRNA, so we have, and you can establish, you can establish these trajectories. Of course, there's many ways of doing this. Here we use Monaco 3 from the Trepno lab. And we know where this trajectory starts. So this is the developmental, the differentiation trajectory, which we of course inferred from the expression of the markers genes before. So I will be referring this as the pseudo time, going from the beginning towards the end. Now, because we have these two modalities of the same cell, what we can do is to look at those genes that are differentially expressed across all these different cell types, and then see how that ties in with the changes in spatial positioning of those genes along this trajectory. So what we've done is there were about 500 genes that are developmentally differentially expressed, of which here about half of them are plotted. And what you're looking at here is the pseudo time. So we plot the expression state along the pseudo time. For example, these genes are expressed early on in this trajectory, and then they are turned off. Whereas these genes are turned off and then they become expressed later on in these neural types, right? So we have the transcriptional dynamics here, but of course, we also have the lab positioning or the spatial genome positioning, which we can now overlay with this. And what we observed is a very close association between gene activities and changes in lab. So for example, for these genes that are on early on here, there's no spatial positioning at the nuclear periphery. So these genes are in the middle, as you would expect from the electron micrograph as well. And then as these genes are turned off, they associate with the nuclear periphery. So as opposed to these genes, which are off early on in this trajectory, they are associated with the lamina in general. And then when they are turned on, they are dislodged from the lamina to allow transcription to occur. So we can now, with this type of system, we can really start to see, or at least better dissect what is cause and consequence. And I'm just showing you just examples of the things that we're doing at the moment. This is by no means systematic analysis, but it's just to give you an impression. So I just plotted 2 genes here, or just the genes are plotted here, where again, you look over pseudo time of the activation of the expression of a gene here and here in blue. And this is one of those genes that are active early on in this trajectory and then becomes turned off. And because we also have the corresponding lab positioning, we can plot the lab contact for this particular genes. And we see that there's very nice correspondence between this gene moving towards the lamina and this gene being turned off. I mean, here we imply that the attachment occurs slightly before the activation, but of course we need to do that much more systematically, but we at least can see that there's a very close association between the two. The same for detachment. So this is a gene that's only active later on in the neurons. And there we see that the detachment occurs much prior to the activation of this gene. So this suggests, but again, this is just a suggestion that you need to detach from the lamina in order to prepare genes for activation. So we're doing this now much more systematically. But this was just to show you an example of the approaches that we take. And take-home message here is that this DamID and T enables inferring relationships between changes in gene activities and LATs in complex tissues. What's interesting to note is that for about half of the genes, so I told you there are 500 genes that are differentially expressed, half of them have a corresponding change in spatial positioning, so which implies that spatial repositioning is important in regulating gene activities in whatever form. And it appears, but more experiments are needed, that in developmental cortex, lamina context change prior to change in gene activities. But this we're investigating currently further. All right, so no, this I'm not going to talk about. I don't have time. So to wrap up, I wanted to show you the mouse system that we developed in close collaboration with the Mundlos lab in Berlin to really look into how the genome, how spatial genome positioning is important in regulating gene expression across different tissues and developmental stages. So for that we need to develop a conditional mouse, and I'll just go through it briefly, but we can conditionally express DamLamB1 in any tissue of the mouse. Upon expression, the cells that express DamLamB1 become green by GFP. We can select those cells by fax, sort them on the 384 well plate, and then perform DamID and T with it and sequence. So these slides are actually made by Mike, the postdoc that is working on this project, and this is a very close collaboration with Mike and Konrad from the Mundlos lab. It's a great system. It's not leaky. It's rapid and precise induction of this system, and we can now obtain spatial genome positioning profiles of any cell type at any stage of development of mouse development. So we captured DamID profiles from different developmental stages, from E8.5 to E13.5, with an emphasis on limb development, and that's of course the interest of the Mundlos lab. But it's also a very nice system where, again, quite a lot is known about transcription factors that drive expression of the different tissues in limb, and including the enhancer landscapes that are important to drive the spatial temporal expression of those genes. So again, we wanted to understand how lab positioning ties in with the regulation of the genes during this development. But we also took brain tissue, heart tissue, lung tissue, intestinal tissue, and kidney, just to have a nice comparison of gene regulation by LATs in different tissues. So this is what we obtained. Again, a UMAP based on the mRNA. It looks, of course, a bit more complex now what I showed you before with the cortex development, where we have this big blob of the limb, we have the lung, the kidney, the brain. These are ES cells. We have an entire E8.5 embryo, heart development. These are muscles. And collectively, we have over 50 different cell types identified here. And what is interesting now, if we project the developmental stages towards on this UMAP, you can very nicely see the gradients that occur here, where we have the E8.5 embryo, which is over here, with projection towards the cardiomyocytes and these endothelial cells, but also projection towards the limb development. You have the E9.5, E10.5, 11.5, and 13.5. So we can very see the development in time in our UMAP. Now we used quite strict selection criteria to select for the DamID cells that pass thresholds. We used 10,000 unique reads for the DamID and I think about 750 unique transcripts. And we obtained very nice DamID profiles, high quality DamID profiles for all the tissues. And you can already see here, just from the example from ES cells to endothelial cells, that we start to see very interesting differences as this domain here that's associated with the lamina and ES cells and not in these cells. Here's the domain that's associated with the periphery and epithelial cells and not in ES cells. Of course, we can do this now systematically for all the 50-something cell types that we had. And what we observe is that there's a lot of variability occurring. So there's a lot of spatial repositioning across these different cell types. And that's about 1/3 of the entire genome. There's also the other third are these CLADs, as I referred to earlier, which are these constitutive LADs that you find positioned at the nuclear lamina across all the cell types that we looked at. And then there are, of course, regions that never are associated with the nuclear periphery. You can also make a heat map just based on the LADs themselves. So you call LADs and you cluster heat map. In this case, the LADs that are positioned close together show a coordination in their repositioning across the different tissues. And then you can, and as we can illustrate here further, so for example, the regions here are CLADs, so across the different tissues, these regions are associated with the periphery, which is in dark red. Then we have 1/3 of the regions that are associated with, not with the periphery, so they're always positioned away from the lamina. And then we have everything in between where we see tissue-specific repositioning of the genome. And now we can see how this corresponds to change in gene expression. Because we have these two modalities, we can very nicely now look at the same analysis as I showed you for the cortex, but now across different tissues. And again, we also have some brain development here, which is now E13.5. And I'm just going to show you some examples of gene expression, just to give you a feel for the data. So I'm almost done. So here's a gene that's expressed only in these progenitor cells in the brain, in these neurons. And we can look at the positioning of this gene in relationship to the nuclear periphery. And we see a very nice direct correspondence with nuclear positioning in that the cells in which this gene is active, you do not see it associated with the nuclear lamina, and it is associated with the nuclear lamina in almost any of the other cells and tissues that we profiled. So again, a very close association, very similar to what we saw before. Also this tintin gene, which is very important for muscles, so you find it only in muscles. So these are the cardiomyocytes and the skeletal muscles. It's only expressed over here. Now, if you look at the corresponding positioning of this gene, you can see it's repositioned away from the lamina, mostly here and here where this gene is expressed, and it's associated with the lamina, almost all of the other tissues. So again, very close association. But then the surprise came actually when we started looking at limb development. And I'm just showing you one example of a very important homeobox transcription factor, SHOX2, which is very tightly expressed only here in the limb. Remember, this is all limb tissue over here and here. So from what I showed you so far, you would probably expect that this gene is lamina associated everywhere except here, but it's actually not the case. So when we look at lamina association, we see that this gene is off the lamina across all the limb tissue, irrespective of its transcriptional status. So it's off indeed there where it's transcribed, but also in other tissues where it's not transcribed. And you can see that it comes off the lamina already in E9.5, and it continues to be repositioned from the lamina prior to this gene being transcribed. So this is a very different scenario where actually transcription activation is uncoupled from the repositioning of the nuclear lamina. Here, repositioning precedes the activation of this gene. Let me skip this because I think I'm running out of time. I'll just go to the summary, where what we're seeing is that where I showed you before, that there's a very tight association between gene activities and positioning for the limb development. We see actually that at E8.5, this important limb gene is associated with the lamina, but then much prior to the activation of this gene, this regions come off the lamina, including actually its enhancer. So this is in fact a whole TED coming off the lamina, including the enhancer landscape, but without corresponding activation of transcription. And then only in the tissue where it's supposed to be transcribed, that's where you see activity of this gene, and not in the other tissues where this region is also dislodged from the nuclear lamina. So of course, this comes with a number of questions. So we can conclude that the lamina associations can be dynamic and cell type specific in development. We see both coupled and uncoupled detachment from transcription. And in the limb tissue, we see that we see entire gene come off the lamina, including its stat and its regulatory landscape prior to the activation of this gene. Of course, that brings some questions in what is now preventing the transcription activity from occurring here at these stages. When this whole region comes off the lamina, is that maybe another type of histone modifications like K27, or is it merely the absence of the transcription factors in these regions that prevent gene expression from occurring? And then what are the mechanisms that determine this very specific tissue-specific activation of the SHOX2, for example, the SHOX2 gene in only these tissues of the limb? And we're currently actively investigating that. All right, with that, I want to conclude and thank the people. So DamID was performed by Koos and Corina together with Franca. LADS in mouse cortex was done by Corina and now picked up by Pim in collaboration with Onur. This I didn't show actually, but we developed this DamID and sort chick method, which was done by Sami and Pim and Kim in close collaboration with Peter Zeller in the Finale lab. DamID mouse was done by Isabel, Robin, and Sami in close collaboration with the Mundlos lab. And then EpiDamID, of which I showed you one image, was done by Kim and Franca together with Sandra. All right, thank you very much, and I hope there's still some time for questions.
Thanks, Jop. There is actually several questions, and there is time. So I can start with the first one from Irina. Hey, Jop, great talk. From free DAM profiles, we see mostly euchromatin. Why?
Yeah, free DAM is a very good marker for chromatin accessibility. So DAM has an intrinsic affinity for DNA. So like TN5 transposase, it likes to go to open chromatin. So simply because it's visible. So you would get, if you express DAM only in cells, you get beautiful single cell open chromatin profiles and transcriptomics. So you need to tether away DAM from open chromatin, which is very nicely happens when you fuse it to lamin to not see open chromatin. But sometimes this causes some difficulties in that it also gives high background levels that you need to take into account when you work with DamID.
Okay. Then Joy is asking another technical question regarding whether the lamin-fused DAM protein is expressed extrachromosomally or it's fused to the lamin gene in the genome. And he's wondering about competitive binding between wild type and fused lamin for heterochromatic genomic regions.
Yeah, so you can do both. So in the case of the S cells, we indeed tag the one endogenous copy of lamin B1 with DAM. In the case of the mouse, it's expressed from a safe haven locus. So we introduce effectively an extra copy, but it's very, we express it conditionally. So it's only expressed for a few hours. And we've done before, not necessarily for this experiment, we've done before a lot of tests whether the expression of this fusion protein would affect the integrity of the lamina. We've done even electron microscopy and super resolution microscopy, measuring the thickness and the filamentous structures, and we've never seen, at least, that it affects anything. And now with the sort, so the chromatin immunocleavage methods, we do see the same patterns in LADs as we observe in those same cell types with DamID. So we're at least confident that the interactions we observe are not somehow a consequence of the expression of the fusion protein. Okay,
so Andrew is also saying great talk, Job. Couple questions. Do LADs show tight correlation with CTCF cohesin, TAD boundaries in ES cells in vivo models? Yeah, we can start with that one first.
Yeah, so in general, and actually something we're actively investigating further, is that in general, the lat borders correspond with tab borders or compartment borders, and they are rich for CTCF. And the same goes for cohesion components. Yes.
And the second is, have you looked at the dynamics of these domains in ES cells using nuclease deficient DPNL GFP reporter?
Yeah, we haven't necessarily looked at the live cell imaging with this method. So that's kind of the microscopy version of the DamID, where we can illuminate the methylation events as they occur in a cell. I don't think we have done live imaging, but we've done super resolution imaging, and structurally it looks the same as in other cell types. It doesn't look necessarily more more dynamic or more decondensed than what you observe in more differentiated cell types. But we haven't done any live imaging of this.
Okay, one is asking about if there is a role in the nucleolus in the LADs.
Well, the only associations between nuclear lamina and nucleolus that we observe is that LADs that dislodge from the lamina after mitosis, we often find them back at the nucleolus. So at least a proportion of what you observe around the nucleolus as this electron-dense chromatin is the same as what you find at the periphery. It's just that in one individual cell, it's positioned there at this platform, and in another cell, you find it around the nucleolus. We don't know whether there is, not everything is at the nucleolus. We haven't investigated yet whether, for example, the regions that are positioned at the nucleolus, whether their transcriptional level is the same as compared to when the same region is positioned at the lamina, as opposed to regions that do not go to either of those platforms, whether those genes are the ones that are, for example, become more active. But yeah, there's correspondence.
Okay, Genevieve says you reported 50% correlation and brought the explanation in the case where there is detachment but still repression. Are there examples in which non-correlation consists in the gene being attached and though being expressed?
Yeah, there is. We haven't looked into that, but there is. I mean, there is about 10% of the genes that are positioned at the lamina are expressed, but we haven't systematically looked into whether the repositioning of genes towards the lamina, whether that does not result or does not associate with repression, or whether they stay actually expressed. But yeah, I'm sure there will be some exception, but they didn't stand out. We haven't looked into it thoroughly yet. Yeah.
Okay, there is several questions that perhaps you can answer in the Q&A. There is actually, I wanted to ask one last question before we move to Anna, which was about the DamID, which I think it was super interesting, that you can just tell us a little bit about the principles. How does it work?
Yeah, it's a very simple principle, and that, of course, you cannot fuse in order to map antibody histone modification. You need antibodies. You cannot fuse antibodies to them. So what we did is to use these MINT bodies in collaboration with Hiroshi Kimura. So these are these single chain antibodies, which comprised of the variable heavy and the variable light chain that you can genetically encode, fuse to them. So you can express that, and then we can very nicely profile histone modifications. The other strategy is to use reader domains, the known reader domains. And we had to fuse those in tandem to them, very similar to what Tunjai Bobec did previously for his proteomics method. This works really well for a single cell DamID as well.
Okay. Thanks a lot, Jop, for the great talk. And then there is some few questions so you can answer in the Q&A. Yep.